Hey folks, and welcome to another edition of Zog Science. We're talking about regulation again. This time we're going to be talking about osmoregulation, so we're going to be talking about water regulation. So let's talk briefly about plants, and then we'll get into animals. Plants can't move, obviously, and therefore they are osmoconformers. And what that means is that whatever the water conditions are around them, they're going to conform to those conditions. So this can cause some problems, right? Because they can't move around. If there's no water in the area that they're at, they're kind of hosed. There's no water for them, so they're not going to be able to do photosynthesis. They're going to wilt, things like that, right? So this is why it's important that you water your flowers. Why plants grow more when it is raining outside as opposed to when it's not raining outside. I don't know if you've looked at the grass maybe in your lawn, but in the winter it's not growing as much. That's partially due to the fact that there's not enough sunlight. It's also due to the fact that there isn't as much water a lot of times available. In the summers, after a big rain, the next day you might notice that the grass is growing really fast, or in the next few days you might notice that the grass is growing a lot. That's because it's got a whole bunch of water and now it can grow. So plants need water, but they can't move around, so they are osmoconformers. They conform to whatever environment they are in. All right, let's do a quick review of osmosis. Water is going to move from the higher concentration of water to the lower concentration of water. And what that means, they're going to be moving from the hypoosmotic side to the hyperosmotic side. The hyperosmotic side has a higher concentration of solutes, which means that therefore there is a lower concentration of water. And that's going to basically, that's going to be key as we talk about how organisms are able to get water inside of themselves. So osmoconformers are animals that remain isoosmotic, or they are at equilibrium with their environment. This must be in salt water, and pretty much the only osmoconformers are things like cnidarians or sponges, things like that. They are going to be isoosmotic with their environment. They're going to be at equilibrium. Again, this only occurs in salt water. It does not occur in freshwater organisms. We can also have osmoregulators, and these are going to control their internal osmolarity. Osmolarity is just the total concentration of solutes per liter. So they're going to be maintaining that different from the external environment. And they are able to live in a variety of environments, whether that is salt water or whether that is fresh water. Now, osmoregulation in salt water versus fresh water is a little different. The salt water are going to be, you need to get rid of a lot of that salt to make sure that you're maintaining the right concentrations. Whereas a freshwater fish is going to be taking in a lot of salt and then excreting that salt through the urine. And we'll talk about kind of how that process of the urine, how that comes about. Now, there are different types of kind of ranges that you can have that you can live in. For example, you can be either euryhaline or stenohaline. Euryhaline means that you can tolerate both salt and fresh water. That'd be something like salmon, right? So salmon, they live in the ocean, but then they swim up the rivers in order to breed. Or you can be stenohaline, which is that you can only live in a narrow range. So basically, you either live in salt water or maybe you live in brackish water, which is the water found where salt water and fresh water meet, or you just live in fresh water. We can also have organisms that kind of live in some pretty extreme environments. We've got these things called tardigrades, which are also known as water bears, and they can survive for decades without water in this kind of dehydrated state. And then once they get a little tiny bit of water, because you notice that they're very, very small, only a few hundred micrometers in length, they can kind of be hydrated, and then they do reproduction and things like that. They can also survive things like being in boiling water, being in vacuums, or being dosed with lethal radiation. So they're pretty cool and interesting creatures. Now, the environment that an organism is in is going to obviously affect greatly how it is going to deal with either obtaining water or retaining water. So if you look at something like a kangaroo rat, these live in kind of desert environments. They only get a little bit of the water they get from the actual food that they bring in. Most of the water is derived from their metabolism, right? Because what do we know about the process of cellular respiration? Well, it produces water, right? That's one of the byproducts of the reaction. So they can actually produce water themselves from the food that they're eating. And then they are actually not going to produce nearly as much urine because they are trying to maintain their water And they're going to lose a lot of their water through evaporation, which is, it's just a natural thing that's going to happen when you live in a desert environment, right? It's going to be very dry air, so you're going to go through a lot of evaporation. If you contrast that with someone like, an organism like a human, we actually ingest most of our water in a liquid form because we have greater water needs. We do derive some of it from the metabolism, but not as much. And then our urine is going to be where we get rid of the most of our water. And we lose a fair amount through evaporation, but it's just not nearly the same. And that's because, again, we live in a different environment. So the goals of osmoregulation is to maintain proper amounts of water and solutes within the body, and is generally paired with getting rid of nitrogenous wastes. So let's talk about what the different types of nitrogenous wastes are. Well, you've got ammonia, urea, and uric acid. Ammonia is the most toxic, and it requires the most water because you have to dilute it. This is only produced by aquatic organisms such as fish. Ammonia is not very energetic in terms of what you need to produce it. And where this ammonia is coming from is it's coming from the amino groups in amino acids, and it's also coming from nitrogenous bases. And the problem with that is that these amino groups are toxic if they build up too much, so you have to get rid of them. So we've got ammonia, which is produced by aquatic organisms. You've got urea, which is what is produced by mammals, amphibians, sharks. Some bony fish do this as well, but not many of them. It is less toxic and requires less water, which is good because we don't live in water environments. So we want to use something that uses less water in terms of producing it, because we want to maintain our water because we're not surrounded by water all the time. Whereas fish, they have tons of water, doesn't really matter. It is a little bit more energetically expensive. It takes a little bit more energy to produce urea than it does ammonia, but it's not as much as uric acid. And uric acid is the least toxic of these three. It requires the least amount of water. However, it is the most energetically expensive. It takes the most energy in order to produce it. And you can kind of see that just by looking at the structures, right? The uric acid has a much more complicated structure than the urea or the ammonia. So it makes sense that it'd be more energetically expensive. These are going to be produced by things like reptiles, which, remember, reptiles includes birds, insects, and then land snails. These organisms, they want to be maintaining as much water inside themselves as they can based on the environments that they live in. All right, so the excretory system is linked greatly with this process of osmoregulation. And there are four main steps to the excretory system. We've got filtration, which is the movement of fluid and solutes into the actual excretory system. Then you've got reabsorption, where water and the desirable solutes, so things that we want to hold on to, are reclaimed. Then you've got secretion, where excess water solute is sent to the filtrate. So, if we've got more water in our bodies than we need, we want to get rid of that excess water. And then finally, we've got excretion, which is where the urine or the modified filtrate, the filtrate is what we're filtering out of the blood, is then expelled from the body. All right, so the vertebrate excretion involves these organs. The main ones are the kidneys and the bladder, but then you've got a couple of ducts in there that connect them. The kidneys is where the actual filtering process occurs and the production of urine. Then you've got the ureters, which connect the kidneys to the bladder. The bladder stores the urine until you get too much of it. And then you've got the urethra, which is where your urine is excreted outside of the body. So the kidneys are where all of the fun happens. And the kidneys are made up of more than 1 million units, which are called nephrons. And you can kind of see how the kidneys are shaped right here. They kind of all surround these areas. And the nephrons are all found inside the kidneys in this cortex. And then there you've got the ureter, which is going to be draining all of the nephrons. So when we kind of take a look at the nephron structure, you've got the glomerulus. Anyways, right here, and that is where the first filtration part of this occurs. And then you've got the loop of Henle right here. The loop of Henle is where the next kind of two portions take place, and then the excretion takes place at the end. So let's take a little bit of a closer look. The glomerulus is where the filtrate enters the nephron at the Bowman's Castle. So you've got some capillaries in here, which are going to facilitate exchange. Then you've got the proximal tubule, which is this right here. The proximal tubule is where the reabsorption of the water, salt, and bicarbonate occurs. The loop of Henle on the descending side is where water is reabsorbed, and then on the ascending side is where salt is absorbed. The distal tubule, which is up here, is where the reabsorption of water, salt, and urea occurs. And then the collecting ducts collects the concentrated urine down here on the very end. Now, you might notice that if you look at both of these blood vessels, these are both arteries, and that's because both of these are still moving away from the heart. They're not actually going towards the heart at this time. All right, so kind of the process of the nephron is a little bit more clear to kind of see what's going on here. So what happens is that you've got kind of these three different layers of the kidney, and each part maintains different concentrations of solutes and water in order to move different materials. So at each spot, basically what we're trying to do is we're trying to concentrate the urea and salt into a final package, and while maintaining as much water outside of the final product of the urine as possible. And so the way that we do that is that we've got different osmolarities, and so as you go down into the inner medulla, you get a much higher osmolarity, and so kind of as you're going through, you've got water that's coming out, and as water moves out of the tubules, there is going to be a concentrating of the urine. Notice that the urine then becomes less concentrated as it kind of comes back up the loop of Henle, and then finally, as it is leaving, it reconcentrates up to the 1,200 level. So, and basically what happens is that we're going to be using active transport. So the active transport of, the active transport of the red arrows is going to maintain a hyperosmotic environment that is going to help to facilitate the passive transport of the water out of the filtrate. And again, the whole goal of this is to produce that hyperosmotic urine. It is not necessarily important that you understand all of the individual steps along the way, as long as you can kind of, and it's not like you need to remember, where all of these things occur. So you don't need to know that in the cortex this happens, the outer medulla this happens. More so, you need to understand the overall idea of what's going on here. The overall idea being that we are concentrating the urea and making urine that is as concentrated as possible. The way we do that is we use the transport of the salt ions in order to produce this hyperosmotic environment so that the water will then move out of the filtrate and back into the kidney itself or into the blood vessels that are surrounding it. Now, there are a couple different hormonal control systems. The first one is ADH. And so what happens is that as the blood osmolarity or the solute concentration in the blood increases, the pituitary gland releases ADH. And the pituitary gland is attached to the hypothalamus, which we've talked about before in relation to the endocrine system. And so ADH is going to increase water reabsorption. So it's going to increase the amount of water that is pulled out of those tubules in order to maintain more water inside the blood, which is good, right? Because you want the blood to maintain homeostatic osmolarity. It's also going to induce a thirst response. So that's going to, tell the animal to begin drinking water, which is obviously then going to help to decrease the blood osmolarity because you're bringing more water into the body. The other system is the RAAS system or the RAS system. Basically what happens is that as your blood volume or blood pressure decreases, the nephrons themselves actually are going to be able to release renin, and there's a specific part of the nephron that senses this decrease in blood pressure. You don't need to know what it specifically is called, but what happens is that this renin causes a cascade, which eventually tells the adrenal glands to release aldosterone, and aldosterone is going to increase water and sodium reabsorption, and that is going to increase blood pressure. One of the kind of molecules along the way for the cascading is also going to cause the arterioles to constrict, and that is also going to increase blood pressure and volume, which is good because you do not want to have low blood pressure or blood volume, because then your blood isn't able to get everywhere that it needs to go. So it's a pretty good system in terms of helping to make sure that you have what you need at all times. All right, that's all we've got for osmoregulation. Hopefully, I'll get to see you all sometime soon. Just make sure that you're being prepared and checking your emails. So, thanks for watching, and I'll see you next time.